Extral hosted a virtual meeting, Advancements in Translational Radiation Research, featuring a panel of leading radiation researchers highlighting translational research done over the last 10 years using the Small Animal Radiation Research platform. The meeting featured 6 leading radiation researchers who highlighted some of their research outcomes using SARRP. Dr. Karl Butterworth from the Queen's University Belfast summarizes the focus of their Translation Radiation Biology Research program.
So broadly speaking, over the past several years since we've had our SARRP, we have been trying to develop a number of different key areas which really, as we've heard from some of the speakers today, are very similar to ongoing efforts around the world. So I think, first of all, we've been developing a number of different drug radiotherapy combinations. We have had historical interests in the application of high atomic number metal nanoparticles as radiosensitizing agents. Professor Berbico is also very active in this area. We're also interested in combining radiotherapy with molecular targeted agents, particularly agents such as inhibitors of DNA damage response, chemokine signaling, and also epigenetic therapies. And then more recently, we have emerging interests in the repurposing of drugs and also in tumor metabolism. The second area of interest that we've been working in really follows the implementation of some key radiotherapy technologies, such as the application of injectable fiducial markers for preclinical applications. We have some ongoing and early interests in the development and application of advanced imaging for preclinical applications using radiomics, and then also in novel aspects of radiation dosimeters, and also trying to optimize radiotherapy based on very complex and novel beam delivery methods such as spatially fractionated radiotherapy, grid and lattice type of approaches. Final area then that we've been working in is really trying to model some clinical trials and reverse the translational research process. So trying to take clinical evidence, develop new models that we can establish in the laboratory, and then take them forward to gain some mechanistic insight and critical information as to what's going on that couldn't be possible in trials in patients. And these are some of the key areas, looking at substructure avoidance, a number of local trials for SBRT and prostate cancer, and then also importantly, trying to model some different patient comorbidities that may have significant impacts on radiotherapy response.
Dr. Butterworth continues and presents some of the future opportunities for radiation biology research.
So I think potential opportunity is to establish SARRP at the center of a robust phase testing platform for drug radiotherapy combinations. This will require, of course, a portfolio of translationally relevant disease models, optimizing delivery methods and schedules with appropriate response monitoring using clinically relevant techniques. In addition, I think there's also possibility to explore novel radiobiological paradigms based on optimized dose delivery parameters. We've seen a lot of interest around optimizing spatial dose delivery using a number of different techniques, some of which are beginning to translate to the clinic. We've seen really tremendous innovations from Professor Jimenez's group in terms of the application of SARRP beamline to study the radiobiological effects of protons, flash radiotherapy, and then also proton flash. Using this image guidance, we've been able to look at critical substructures within target organs of risk, which really wouldn't have been possible using conventional approach. Finally then, I think there's huge opportunity to integrate SARRP, to continue to integrate SARRP with novel radiotherapy and imaging technologies from both the physics perspective of automated segmentation, treatment planning optimization, image analysis, and online adaptive radiotherapy, and then also combining this with molecular and functional imaging to biologically optimize and target specific regions within tumors and tissues.
For more information on the Small Animal Radiation Research platform, or to learn how SARRP can help you with your research needs, visit extral.com/SARRP.